Why am I muted? Am I unmuted now? All right, so the cytoskeleton serves as the cell skeleton, microfilaments, intermediate filaments, and microtubules. Of course, you see this as microvilli, and of course we use that for absorption, whereas we use cilia for movement across the surface of the cell. The smallest cytoskeletal structures are the rod-shaped microfilaments. These protein strands are generally about 5 nanometers in diameter. Typically, microfilaments are made of a protein called actin. And when we get into actin and myosin, we will talk about that is muscle contraction. Intermediate filaments and microtubules. I think we covered this a little bit before in the last video. Intermediate filaments varies among cell types. These filaments range from that size. They are so named because of the intermediate in size between microfilaments and microtubules. What do? They strengthen the cell and help maintain the shape, stabilize positions of the organelles, and stabilize the position of the cell with respect to surrounding cells. Microtubules, a globular protein called tubulin. Microtubules are the largest component of the cytoskeleton. They extend outward into the periphery of the cell from the region near the nucleus called the centrosome. And again, microtubules have the following function: maintain portions of the cytoskeleton, giving the cell strength. Microtubules change the shape of the cell and may assist in cell movement, and microtubules can serve a kind of monorail system to move, which is what I talked about through moving stuff from one side of the cell to the other. During cellular division, microtubules distribute duplicated chromosomes containing the DNA, and that's where we talk about the spindle apparatus, and it attaches to the DNA, the replicated DNA, and pulls them apart. So each cell gets some DNA. Okay. Centrosomes and centrioles, there they are. Okay, we talked about this already. Here's your motile cilium, ciliary movement. Okay, this is describing the array that it's set up. We don't necessarily need to know the array and how it's lined up or how it's set up, just factor what it is and how it works. Now remember, okay, we have things that are motile, so we have cilia and flagella, and in the human, sperm are the only flagella, but we have lots of places for cilia. Okay, so we have primary cilium is found on cells of a wide variety of tissue body. The structure is similar to that. The non-motile primary cilium acts as signal sensors. So these are sensors as well as movement. Sensors as well as movement. The motile cilia move things across the surface, like we described that before. Then, of course, we have the flagellum there, which is the sperm to fertilize the oocyte. Ribosomes, we talked about the ribosomal subunits. We talked about the protein synthesis, free and fixed ribosomes, which are right here. The free ribosomes are scattered throughout the cytoplasm. This is for this particular cell, and the fixed ribosomes are on the rough ER, where it allows that newly synthesized protein from the ribosome to enter the ER, where it is then shipped to the Golgi apparatus. Proteasomes are organelles that contain the assortment. Okay, cytoplasmic enzymes contain attached chains of ubiquitin, a molecular tag of 2 proteins destined for recycling. Tagged proteins are quickly transported into the proteasomes. Once inside, they are rapidly dissembled into amino acids and small peptides, which are released into the cytoplasm. The endoplasmic reticulum, ER, is a network of intracellular membranes continuous to the nuclear envelope, so it attaches to the nuclear envelope, which surrounds the nucleus. The name endoplasmic reticulum is very descriptive. Endo means within, plasma refers to the cytoplasm, and a reticulum is a network. The ER has the following major functions: synthesis, storage, and transportation detoxification. And that's both of them. That is the rough and the smooth. Okay. So the ER forms hollow tubules, flattened sheets, and chambers called cisternae. 2 types of ER exist: smooth ER and rough ER. The term smooth refers to the fact that no fixed ribosomes are associated with smooth endoplasmic reticulum, while the fixed ribosomes on the outer surface of the rough endoplasmic reticulum give it a beaded, grainy, or rough appearance. Again, we said before that the rough ER was protein. The rough part is the ribosomes, which actually create proteins, right? And then it sends it into the inside the cisternae, which is part of the ER. And then, of course, it then ships it out to the Golgi apparatus through vesicular transport. Remember, a vesicle or vesicular transport is nothing more than a pouch. A pouch. Next, we have the smooth ER, which is a synthesis of phospholipids and cholesterol needed to maintenance and growth of the plasma membrane. Remember, we talked about smooth lips, and there it is right there, lipids, okay? So the smooth ER, not only does it do detoxification, but it also creates the phospholipids and cholesterol needed for maintenance of the plasma membrane. And before we said, what is the plasma membrane? It is a phospholipid bilayer with the cholesterol on the inside for structural support. So, but we do have to replace that. It does have to be regenerated eventually, okay? A synthesis of steroid hormones such as androgens and estrogens, the dominant sex hormones, in the reproductive organs, okay? So that's what the SCR does, the smooth ER. Synthesis and storage of glycerides, especially triglycerides, in liver cells and fat cells, and synthesis and storage of glycogen in skeletal muscle and liver cells. Okay, so that's the smooth ER. So here down at the bottom of the rough ER, you see, okay, so it modifies and packages a newly synthesized proteins. And then these transport vesicles then deliver their contents to the Golgi apparatus. Again, we said Golgi apparatus is FedEx, shipping packaged proteins, whatever that might be, through secretory vesicles out of the cell. And then, of course, don't forget, we also have the lysosomal enzymes or digestive enzymes that in vesicles, which are created from the Golgi apparatus, and those proteins that are coming into it. All right, so here's a nice protein synthesis. Now remember this outlook, okay, because you come back here to talk about more of this stuff here. Okay, so this is actually an important spotlight. So what we have here is a cell, obviously, and there's our phospholipid bilayer and another bilayer here on the nuclear envelope with the nuclear pores. And we'll come back to this and talk more about transcription and translation in a bit. So here's the DNA, here's the nuclear pore and the nuclear envelope, the chromatin, and remember chromatin is uncoiled DNA. This is all the nucleoplasm, and don't forget we have a nucleolus inside here. Okay, so here's the DNA, uncoiled, doing its job. If it's coiled up, and you see that normal X that you see, everyone shows you chromosomes and how they look like that X, well, that's coiled DNA. It's coiled around histones to make it nice and tight so that we can go through the replicative process or mitosis. Okay, so then this is the DNA. It unzips at a certain portion, and it knows where to unzip to create messenger RNA. So it unzips at a very specific spot. It creates those, remember that complementary base pairing, A, G, C, and T? It creates a messenger RNA complementary from this start part to this end point. Okay, and then this section of messenger RNA is a representative of a protein. This mRNA messenger RNA leaves through the nuclear pore. Now this pore is so small that the DNA cannot fit through here, so messenger RNA is the only one that can. Once the messenger RNA leaves, it actually then will go to either the free ribosomes or the fixed ribosomes. Now it can go to either one. Okay, it can go to either one, and then it starts creating, using the messenger RNA, it starts creating these amino acids, start creating chains of amino acids, and that's, of course, remember what we said before, is what's the building block for proteins, which are amino acids. So it attaches these amino acids through peptide bonds and creates these chains, which are then proteins. Okay, so now, of course, this free protein from the, this protein from this free ribosome is for this cell. And then the fixed ribosomes on the ER actually send it into it, where the rough ER then modifies it. Then it pinches off a little vesicle, which transports it to the Golgi, which then does, as you see here, digestive enzymes or lysosomes, lysosomal enzymes, or it's a secretory vesicle, which then releases it through exocytosis out of the cell. Okay, and of course, look at all these numbers up here of 1 through 8. These will help you describe what's going on here. Okay, so good. We'll come back to this when we talk about transcription and translation. Okay, it makes it a little easier to understand that. All right, so there's your lysosomes, primary and secondary lysosomes. It's just talking about how it attacks things, and then the damaged organelles it destroys, and then it releases them. Okay. Peroxisomes are smaller than lysosomes and carry different groups of enzymes. In contrast, lysosomes, which are produced by the Golgi apparatus, new peroxisomes are produced by the growth and subdivision of existing peroxisomes. Their enzymes are produced at free ribosomes and transported from the cytosol into the peroxisomes by carrier proteins. All right, so look, oxidative enzymes or peroxisomes, oxidative enzymes, they break down things like hydrogen peroxide. Okay. Okay. Mitochondria. Okay. All right. So the cells of all living things require energy. So remember we said before that the mitochondria actually produces ATP, which is adenosine triphosphate. This has a high energy bonds, right? High energy bonds that allow for, when we break that bond, the energy is then released. Remember that potential and kinetic energy stuff? Okay. So the ATP is potential energy, which then breaks that inorganic phosphate off. And that bond then, once it's broken, releases energy, and that's the ability to do work. And then, of course, we use that energy to do things like muscle contraction or whatever. Okay. Now, so remember what I said before, we have to talk about, when we say talk about the mitochondria, ATP production, and if here you look at you see this oxygen comes in, okay? So if you look down at this little drawing here, okay, you have to have oxygen to get into the mitochondria. All right, this is called aerobic respiration inside the mitochondria. All right, so as glucose comes in, we talked about glucose, which is C6H12O6. Glucose is the energy, right? So as it comes in, starts to come into the mitochondria, it's split in half. So C6H12O6 is split into C3, C6H12, so H6O3. So it's split in half exactly, right? And then we end up with pyruvate, 2 molecules of pyruvate, and it can only enter into the mitochondria if it has oxygen. This is aerobic respiration, which is called the citric acid cycle. This is how we make our ATP. Okay, this is how we make our ATP. If we don't have oxygen, in other words, let's say we're running a marathon or whatever reason, we're in a deprived oxygen area on a mountaintop, whatever, you will start creating, you'll go through the glycolysis. We always go through the glycolysis process, no matter what, because we still go through this process and it still makes this byproduct. But primarily we get into this inside the mitochondria. But if we can't make get into the citric acid cycle, we go through glycolysis, and the byproduct of glycolysis is called lactic acid. All right, now listen, when we get into the muscle contraction, we'll talk about this a lot. But if we make lactic acid, it's called an oxygen debt, or we actually have to pay that back. So when we make lactic acid, say we're exercising beyond the ability of our body to get oxygen out to our tissues, we create this stuff called lactic acid. If we have to get rid of that lactic acid, and we do that by keep continuing to breathe faster. So this is why when you exercise, your metabolism stays up, because you're continuing to breathe faster to pay that lactic acid, get rid of that lactic acid. Okay, all right, so anyway, so remember, in the cytosol, remember this is outside the mitochondria, but inside the cell. So the cytosol, which is the fluid inside the cell, this is where we have anaerobic respiration, and without anaerobic respiration. Inside the mitochondria is where we have aerobic respiration. Okay, and this is the citric acid cycle, which has other names, which we'll talk about when we get into the muscle tissue, which is citric acid cycle, the Krebs cycle, or the tricarboxylic acid cycle. Okay. All right, so there is a lot of information through that process right there. And of course, that what I just talked about right there is all this right there. Here's that tricarboxylic acid cycle. So if you didn't understand what I was saying, these are all the same thing. Citric acid, Krebs, or TCA. Okay. And then of course there's your aerobic metabolism or cellular respiration, aerobic metabolism, aerobic respiration, same thing. Okay. Okay, so membrane flow or membrane trafficking in an actively secreting cell, an area of equal to the entire membrane surface may replace each hour. This is as things are going in. This is what membrane traffic is. This is things coming in and leaving the cell throughout the day, throughout the hour. Okay. We'll talk about different ways that that actually occurs later. I think it's coming up actually. All right, so the nucleus contains DNA and enzymes essential for controlling cellular activities. Okay, so the nucleus, we definitely can see the nucleus when we stain it in cells. The structure of the nucleus, and of course we talked about the structure of this. So nuclear pores, nuclear envelope, chromatin, nucleoplasm, nucleolus, control center for the cell. Contained the chromatin is uncoiled DNA. This is the everything that we have that describes everything about us. All right. Okay, so again, the nuclear pores allow the messenger RNA to release. DNA coils around histones. So when these histones, this actually helps the DNA coil up around, here it is right here. So this is what I was talking about before. This is that X, okay, the X. So sister chromatids, these are chromosomes, these are chromosomes, which is your DNA coiled up around each other. And when you see this in the X format, this is actually the original DNA and a copy of the DNA. So one is going to go to one cell, the other side is going to go to the new cell. And then remember those centrioles, they come down and they attach right here to the kinetochore, right, and actually pull apart. So those microtubules, remember from the centrosomes, pulls this side this way, pulls this side this way, and then separates the cell. So we end up with this, what we call a metaphase plate, where all these chromosomes line up, and then it pulls it apart. Okay. Anyway, that's the histones. Rolls it up, tightens it up. Okay, so genetic code. How many, do you guys remember how many chromosomes we have? Right, 23 pairs, right? Mother and father, 23 pairs, 23 pairs. Remember that commercial 23andMe? That is for your chromosomes. That tells you all kinds of stuff, you know, your heritage, your allergies, all kinds of stuff. All right, protein synthesis, transcription, and translation. Okay, so we have gene activation that will start something to create proteins for whatever reason, whatever it is. So transcription is the synthesis of RNA from DNA template. Okay, so that's just the definition, right? So let's talk about what it is. Okay, you do not have to go through the process and how to actually go through and make transcription or make messenger RNA, but here it is. Okay, so remember, complementary base pairs into RNA. Remember, U in place of what? U in place of T. So in RNA, T will become U, right? Okay. All right. So this is making the new messenger RNA, which is the messenger RNA is the basic code to make a protein. It is not a protein yet. It is just rmRNA. Okay, so this is transcription. Where is transcription occurring? This is inside the nucleus. Okay, inside the nucleus, in that chromatin. Then we translate that messenger RNA from a messenger RNA format into amino acids or into proteins. Okay. All right, so it converts it into amino acids, which then we attach those amino acids together through peptide bonds, All right, and then those peptide bonds, of course, attach those amino acids, which then is our building blocks for proteins. So let's go to, here's that amino acids and attaching amino acids, and then you see the peptide bonds, and these guys bring down each amino acid, and we end up with this protein. So messenger RNA, and then we end up with this protein coming up here. All right, so this particular anticodon fits this one, which carries this amino acid, so it brings it down to attach. Bring another one comes down for the anticodon. Remember, opposite, and then it brings it down. This is the transfer RNA. Okay, so let's go back to, I got to go back to this picture. So now knowing where transcription and translation occur, or what they are, so we're making proteins, right? That's the job of the nucleus. When you say, what does a nucleus do? Yeah, you could say control center, but more accurately is, oops, one more back. More accurately is what? Protein synthesis, right? Okay, so protein synthesis. That's the big deal with the nucleus. It is protein synthesis. And I think we're down here. Let me come over here. Spotlight, there it is. Okay, so let's talk about what we just saw there with the, here it is. Here is transcription. So first thing you have to do is you have to write it down. Okay, first thing you have to do is write it down. What do you write it down at? Messenger RNA. Okay, so this is transcription. This occurs inside the nucleus. Okay, then what happens is that messenger RNA leaves through the nuclear pore, goes and attaches to free or fixed ribosomes. Then you see it looks like a ticker tape, right? So, but this is, remember, this is messenger RNA. So we convert from messenger RNA into amino acids, which then bond the amino acids together to make proteins. Remember, the building blocks for proteins are amino acids. Amino acids are held together by peptide bonds, okay? These bonds hold these amino acids together. Together, all these things make a different shape, okay, which is a quaternary structure, which is a protein, okay, which is a protein. Okay, so transcription, and this is translating messenger RNA into what? Into amino acids into a protein, okay? So transcription occurs in the nucleus. Translation occurs where? In the cytosol, right? In the cytoplasm, cytosol, it's a liquid. Okay, all right, so remember that, all right? So then those newly formed proteins in the rough ER get modified and packaged, shipped through vesicular transport to the Golgi apparatus, where it's modified, stored, and shipped out of the cell, or it creates lysosomal or degradative enzymes, digestive enzymes. Okay, so let's close that one, go to the right, where we were at before. Okay, come on, computer. There you go. Okay, so we talked about there's transcription and translation. Okay, again, I don't want you to go through this other than to understand where you're at, what you're doing. So if I say, where does transcription occur, or where does translation occur? Where do we write it down at? And what are we writing it down as? We're writing it down as messenger RNA. And if you're doing that, it must be inside the nucleus. If you're translating that messenger RNA, you're in the cytosol of the cell. Cytosol of the cell. Okay, so some definitions, things there. Look at that. There's transfer RNA, anticodon. Make sure you understand those. Here it is, the translation that occurs in the cytosol. Okay, now let's keep going here. Make sure you read the DNA stuff. It's just will help you understand a little bit more. I'm not going to make you learn how to make DNA. I'm not going to make you learn how to do transcription, translation as far as the component level of that, but other than what we just talked about. Okay. All right. So next we're going to talk about diffusion. Diffusion is a passive process, and when I say passive, I mean it does not require ATP. You'll hear it a lot. Passive process does not require ATP. All right. So diffusion. First of all, let's talk about permeability. And what is permeability? It's the property of the plasma membrane that determines precisely which substance can enter or leave the cytoplasm. Okay, so things come in and out of the cell. They can either come directly through the cell membrane, or they have to go through a protein channel, which we talked about before when we talked about the plasma membrane. if they're coming directly in, they're freely permeable if they can just come right in the cell. All right. And if they can't get in the cell, then it's impermeable. Okay, it's impermeable. But our plasma membranes are called selectively permeable. Part of that property is because they have polar heads and non-polar tails. And the other thing is we have protein channels that allow certain things to come in at given times. Okay, so things are either freely or selectively permeable, impermeable. But our cells are selectively permeable. Okay, so only certain things are invited to the party with selectively permeable. Okay, only certain things are invited to the party. Okay, so diffusion, all right, is the net movement of particles down the concentration gradient. So in other words, for example, if grandma walks in your room and she's got her grandma perfume on, when you smell her perfume, all right, that is diffusing off her body, and then you're breathing it in and smelling it. All right, that is diffusion. You didn't take the little explosions for it to come see you. It just actually wafted off her and then went into your nose, and then that's how you smell it. That's actually diffusion. It's down the concentration gradient. So the concentration of the perfume is with grandma, and then most of it comes off, and then, or a lot of it comes off, and then we smell that. Okay, that's down the concentration gradient, or downhill, as it were. All right, so here they're showing you dropping a color-coded sugar cube into water. So as you drop the sugar cube in the water here, you have this concentration down here at the bottom, and then what do you see here? It's starting to spread out through the fluid, which it's the concentration is down here, but it's moving away from that. This is down the concentration gradient, and then until we get to an isotonic solution here. Okay. All right, so that is an isotonic solution. Okay. All right, so some things that affect diffusion rates. Remember, it's a passive process, does not require energy to do this. So distance, the shorter the distance, the more quickly concentration gradients are eliminated, which makes sense, right? Ion and molecule size, so the smaller the ions and molecule size, the faster rate of diffusion. Okay. Temperature, so you increase the temperature, it moves faster. And then how much of the stuff you put there, so the concentration gradient, how much you have there. And then, of course, electrical forces. Opposite electrical charges, positive and negative, attract each other, and like charges repel each other. Okay, so the cytoplasmic inner surface of the plasma membrane has a net negative charge relative to the extracellular or outer surface. Then this negative charge that tends to attract positive ions from the extracellular fluid into the cell while repelling the entry of negative ions. Okay, that's a nice little tidbit. We talked about before how sodium likes to be on the outside and potassium is on the inside. Yes, although they are both positive cations, there's a higher positive charge on the outside and a more net negative charge on the inside of the cell, so it actually attracts quicker, which is a reason why we have when we get into anatomy 2, we'll talk about the heart contraction and how this plays a role. That's the electrochemical gradient. So diffusion across the plasma membrane. Now, what are we saying the plasma membrane is, right? It's a phospholipid bilayer with protein channels in there, extracellular fluid, intracellular fluid, or cytoplasm with all the stuff inside of it, right? So large molecules can't leave or come out through the plasma membrane, but things that are lipid-soluble molecules diffuse through the plasma membrane. So it has to be lipid-soluble, which means it's what? Non-polar, right? Non-polar. And if it's small enough, like, remember what are some things that we talked about that are non-polar or lipids? They're steroids, which are testosterone and estrogen, can pass right through the plasma membrane and affect this cell through with whatever it's doing. That's a hormone, and that actually affects the cells, and it can pass right through this plasma membrane. If it's non-lipid, it has to go through a channel, and there's a bunch of different ways to get into the cell that way. Okay. Lipid-soluble drugs, dissolved gases such as oxygen, carbon dioxide, and water molecules also enter and leave our cells by diffusing through the phospholipid bilayer. Okay. So channel mediated, this is a special case here for the water. That's what we'll talk about that later. Most of the time it's aquaporins, which are channels for water. Channel mediated diffusion, okay. Let's see, membrane channels are very small passageways by transmembrane proteins, and average channels about that big diameter. While molecules can enter or exit freely. These are the aquaporins. Let's see here. So osmosis, diffusion of water across the selectively permeable membrane. This is really the definition of osmosis. So remember, if it's diffusion, it's a passive process. It does not require energy, okay? So diffusion of water across the SPM, the selectively permeable membrane, is osmosis. So let's get down here real quick. Osmosis is the diffusion of water molecules across the SPM. Osmosis takes place across the SPM that is freely permeable to water, but not to the solutes. In osmosis, water flows across the SPM toward the solution that has the higher concentration of solutes, because that is where the concentration of water is lower. Okay, so let's jump over here real quick. Now look here. So you have a U-shaped tube, and it's filled with water and solutes and an SPM. If you don't have an SPM here, this won't work. Okay, it'll be a different process called diffusion, not osmosis. Okay, diffusion would just, if you took this SPM out, these solutes, which are the big purple ones, would just, you can see there's more over here than over here. You would see this, they would just transfer over until they became an isotonic solution, higher concentration to lower. But with an SPM here, a selectively permeable membrane, that water then will leave across to the other side. It will actually come to this side and cause it to increase on the sides. Now, of course, you can't do this with gravity, right? Gravity would not allow this to occur. It would stay just like this, but that's because we have this here. We actually have a force, a charge, a force that causes the water to come to this side until the ratio of water to solutes is equal. Does that make sense? Until the ratio, this is how kidneys work, okay? So until the solution of water to solutes is equal, okay? Now, we can stop this process by putting a force on this side, which stops the pressure, stops this process, right? And that is called osmotic pressure, the amount of force applied here to stop this process. So pushing against the fluid generates the hydrostatic pressure. Okay, let's move on. So osmolarity and tonicity. The total solute concentration in an aqueous solution is called its osmolarity, okay, or osmotic concentration. So therefore, we often describe the effects of various osmotic solutions on cells. We usually use the term tonicity instead of osmolarity. A solution that does not cause osmotic flow of water into or out of a cell is called isotonic. So the term osmolarity and tonicity do not always mean the same thing, although they are often used interchangeably. Osmolarity refers to the solute concentration of the solution, but tonicity is a description of how the solution affects the shape of the cell. Okay? So if you come down here, this is describing tonicity. All right, but remember, what's the definition of osmotic flow? Selectively permeable membrane. So the flow of water across an SPM to the area of higher concentration or higher tone. Okay, so as you look at an isotonic solution, same amount of water flowing in as in out. Okay, so we have that flow across, right? All right, but a hypo, which means what? Hypo is below. A hypotonic solution means that what? There's less solutes outside the cell than inside the cell. So this is a solution. That means the water is going to flow in. Remember, there's more. That's just saying a hypotonic solution means there's more constant solutes inside the cell. A hypertonic solution means there's more solutes outside the cell, and of course then this cell will dry up as water leaves it. So this is where the cell will swell up, and this is where the cell will dry up. This is called crenation, and if this goes too far, this will be hemolysis, and this is only in red blood cells, by the way. What you see here is a red blood cell, yes? biconcave disc, that's a red blood cell. If you swallow it up, it's called hypertonic, or it's called hemolysis if it ruptures. Now, if it's shrinking, it's called crenation. Okay, it's called crenation, and that's just for the red blood cells. Okay, so make sure you do the mental gymnastics on that, and then of course read your normal saline and solutions and plasma membranes and stuff like that. Okay, that will help with that. Okay, so looks like let me see where my time is here. Okay, so I'm kind of running out of time here. Let me see where we're at. Let's stop at 3.6. That's a lot of information. So I'm going to go ahead and stop the share here.